Hi students, we're just giving you a quick tour of our diagnostic microbiology lab, BacT section. So this is a quick glimpse into the area where samples are collected. They are put together and sorted. We have some that go to the urine section. We have some that will go to what we call C's, which is sputums and swabs and many other types. And let me just quickly show you. This here is a cold room, which is a huge refrigerator, which we use for various things. This is actually an incubator, huge incubator, which we use for various things. This one is for organisms that need air to grow. We have other incubators to give things like CO2, etc. We're going all the way up to our blood culture. This is just one kind of sample, and we're going to show you how it is processed and how we use automation. So this is a biosafety level 2 lab you would have learned. So if you look at the door, you will see that it has a sign to say that it's a restricted area authorized personnel. You'll notice that we have our biosafety cabinet, right? And when you turn on our biosafety cabinet, there is a grill. Light. That, let me piece of paper towel. When air gets in, it's sucked down in this grill. You notice that the grill sucks the paper towel down. So it is sucked down, filtered, and then clean air goes onto our sample. So this is where the sample is processed. Certainly, if there's going to be any aerosolization, you want this happening here. These are our loops. This is our BacT incinerator to flame them to get rid of the bug. Now, when the blood culture comes on, we accession it, check it, enter it into our system, the LIS, which we'll talk more about. And this machine, the BacT Alert, is an automated system that allows us to process the bottle. It's like an incubator, but it's able to read the bottle if there is growth and signal growth, so we can go for that particular bottle and process it. So we'll take it back out. We will grow our bacteria on place, which we'll talk more about. We'll do our gram stain. And on this side, you will see they would have had the plates and they use biochemistry and all other things to do ID and susceptibility testing. This is just a picture of something that grew before, and then it's susceptibility testing on it, just to give you an idea of what things look like. All right,
so what do the blood culture bottles look like, Miss?
Okay, can I open this down?
No, there are some bottles.
Okay, so here are some bottles. These are what they look like. This is one set, color coded. One is aerobic, one is anaerobic. I can't remember which is which right now, but we will teach you that later on. And for pediatrics, we have a special bottle. This yellow one.
All right. So this is a pediatric bottle with the yellow. And so what's the barcode on the side of the bottle for?
Right. The barcode on the side allows us to scan the bottle onto the machine so that it can be specially recognized and it's spot known, so that when it's time to take it off and so on, that can be done. We also generate our own barcode. So there's a barcode for the machine and there's a barcode that we generate to enter the sample onto our laboratory information system on the computer over there, which is an automated system that we use to process samples. So all of the reading of the plates and the resulting is done on the computer through our laboratory information system. It makes the system more efficient, and so we're able to get reports, send them out to the ward without having to print them and so on real time.
So when you scan the bottle, does the patient information go on, or do I have to type it all in?
Well, it depends. Now, there are some systems that allow for, at the time of the request, for the information to be entered, and then the sample comes with the barcode. So when we scan it, everything goes on. Where we are now, we are the ones that enter it. So the system also allows for flexibility, so we can enter it, or if there was something inaccurate, we can go on and change it and so on.
So Miss, can we see inside there
Miss, can you open one of these? Any one of these? Okay. So this is the incubator. It's really like a drawer, and it's at the right temperature and giving the right sort of atmosphere. And each well is for each bottle, okay? And it's going to be incubated, and we'll talk about in the lab how if there is growth, there's CO2 generated that changes the reflectance at the bottom of the bottle. Right, it changes the appearance here, and so we get a reading. The machine is able to read it, and on the screen here will tell us if it's positive and which draw is positive, and then we can go on.
So Miss, if I pull out one, can I just stick it in at any other slot that I want to?
No, you cannot. Good, because you don't want to make any errors, right? Now,
so each patient's bottle has its own
specific
spot.
slot.
Okay.
Now, in the past, manually, we would have to observe every single bottle ourselves every day, sometimes needle it and sample it. You can imagine that it's quite tedious. Just want to show you quickly one more machine that we use to help automate the process of processing samples. And this machine is used to identify organisms and do sensitivity testing. So this is the Vitec machine. Okay, it's not on right now. We're not using it right now. We'll talk more about it in the lab, but it can be used. We put the organism on cartridges. We don't have any examples now, but we put them in on these trays, and we put them into the machine, and the machine is able to run the various tests which will be in the cartridges and susceptibility.
Let's see if we have any cartridges.
No, you guys don't have any. No.
Not at all.
Girl, you're brave.
Usually there's one that I can use.
And this, of course, is our monitor. One of the newer automated instruments, the matrix-assisted laser desorption ionization time-of-flight machine, also mainly used for identification, but can also be used to some extent for susceptibility testing. And that's all we have, folks, for you today.
And all these machines are connected to our computerized system, Miss?
Well, right now we have 2 of them that are, and we're working soon to have this one added. And so all of them will feed information into this one space where all the results are collated and interpreted, and reports are generated for the staff and the patients.
So Miss, you showed us a walk-in incubator. What's this?
Okay, so this is also another type of incubator. This one is our CO2 incubator. Okay, and what this does is it gives you the right amount of CO2, 5%, that is needed for organisms that need CO2 to grow. We call them capnophiles. There are some incubators that can generate anaerobic conditions. Otherwise, for the obligate anaerobes, we use jars, okay, that will have gas packs in them. I'm looking to see if we have any. We may have to go in the other incubator. And we put the plates in those, and then the anaerobic conditions are established there, and the organism will grow.
Okay.
Thank you, Dr. Reynolds, for your excellent camera work. That's all we have for you students today.
Thank you, Miss.